Today's Wednesday. Here you go, Sarah. There we go. That might work out.
Alrighty, good afternoon, my friends. Oh, come on, good afternoon, my friends.
That's awesome.
Alright. Okay, so let's review a little bit of what we talked about on Monday. So we started talking about specifically those substances which are found in living organisms, specifically the human body, right? On Monday. We said that let me take a step back. We started talking about water, the properties of water. And we said the properties of water, that those properties that allows water to help sustain life, the reason why we are 60 to 80% water, right, are caused by the attraction between different water molecules, which are called what? Hydrogen bonds, right? Hydrogen bonds is the force between the slightly negative end of oxygen and the slightly positive end of hydrogen on two separate water molecules, right? Those are called hydrogen bonds, right? Hydrogen bonds can only form between water molecules because they are polar molecules. That is, the hydrogen and the oxygen within a molecule share electrons unequally. And so you get a slightly negative charge on the oxygen and slightly positive charges on the hydrogens. And so the slightly negative end of one water molecule is attracted to the slightly positive end of another water molecule, and that is called the hydrogen bond. Right? And so the fact that one water molecule is attracted to another water molecule gives water some unique properties. What are some of those properties? Surface tension, right? And that's caused by water molecules being more attracted to one another than they are the air, right? In fact, it's the cohesion, or the cohesive properties of water that's responsible for surface tension. Water is more attracted to itself than is the air. What's another? High heat capacity. Water can absorb a lot of heat without changing temperature, because in order for the temperature to change, these water molecules have to move away from one another and move faster, right? So that's why we use water to cool things off, that sort of thing. What else? Right, it acts as a cushion, right, for absorbing a lot of mechanical energy, amniotic fluid, the cerebral spinal fluid, right, helps protect your central nervous system. Good. Others? No. Then also chemical reactivity, right? It can participate in dehydration, synthesis reactions, where water is given off and smaller molecules are combined with a larger molecule, and the opposite, which would be hydrolysis, where a water molecule comes in and breaks a large molecule down into smaller molecules. Right? That's basically what happens during the process of digestion in the GI tract. Right? What are called the hydrolysis reactions, as water comes in, breaks bonds and cells. Okay, cool. So then we start talking about organic compounds, right? Organic compounds. So what's a property of an organic compound? It's what? Organic compound? This is all right. Let's say organic compound. Okay, what? So it has carbon. Has carbon. All right. What else? Tends to be big, right? And someone. Right. Most of the bonding is usually covalent. Okay. And in general, what produces organic compounds? Living organisms. Right, living organisms. Lee, what's up? What do you want to say? right? Well, it's composed of carbon, right? And you're correct. They are composed of. Well, later we'll talk about polymers, right? You can have a small unit of it, and then you can attach. You can combine small units to make larger units. Good God, right? We're at war again.
Wow.
Okay. Right. Okay. Cool. And the first organic compound we talked about was what? Carbohydrate, which literally means hydrate or carbon. We said that carbon, hydrogen, and oxygen are in a 1 to 2 to 1 ratio, right? Literally means hydrate of carbon. Okay. And where do we get carbohydrates? Where do we get our carbohydrates?
Sugars and starches.
Well, that's what they are. They are sugars and starches. I should have said. That's what I first thing I should have said. Carbohydrates are sugars and starches. All right. And we consume carbohydrates. Right. That's how we. That's what we do with carbohydrates. Okay. We consume them. Right? Where do we get our carbohydrates? Don't say from food. I mean plants. Right. Sugar beets, high fructose corn syrup. All of the starches that we consume, right? They come for the most part from plants. So you get a little bit from the meat that we eat because there is some glycogen in skeletal muscles, that sort of thing. But for the most part from plants. All right. So you get them from plants, and your body uses carbohydrate almost exclusively as what?
Short-term energy.
Energy. Right. Energy. Short-term energy. Right. You consume a sugary meal, right? Potato chips, bread, your body is basically, it's either going to use that as energy, or if it doesn't need it right now, it's going to store it as energy, right, to be used later. Very little is incorporated actually into tissues. There's a little bit, but not much. Okay? Okay, and we said we classified carbohydrates by their size, right? Okay, so the smallest unit of a carbohydrate you can have is called a what? Monosaccharide. Monosaccharide, right? And if you take two of those monosaccharides and combine them by a dehydration synthesis reaction, you get a disaccharide. Cool. And if you keep combining bunches of monosaccharides, you get a long polymer called a polysaccharide. Oh, by George, I think they got it. All right, give me an example of a monosaccharide. Glucose, all right, blood sugar. Disaccharide. Sucrose, right? Sucrose, which is table sugar, or lactose, right? We're from Wisconsin. Everybody should know that, right? Lactose. And what about polysaccharide? Cellulose or glycogen, right? All the when they talk about complex carbohydrates, that's what they're talking. All right, cool. Okay, I think we finished pretty much talking about carbohydrates, right? That's where we ended. Okay. Right? Okay, so I want to pick up from there. That's the first organic compound we talked about. Now let's talk about lipids. Okay, lipids. Alrighty, so when you look at the structure of a lipid, or I should say if you look at the components of a lipid, it consists of, like carbohydrate, carbon, hydrogen, and oxygen. However, the carbon and hydrogen atoms greatly outnumber the oxygen atoms. Also, lipids are insoluble in water. Give me an example of a lipid. Somebody, raise your hand. Let me call it. Olive oil. You said butter, right? Right. She said Susan says parquet. What? Eggs. Well, there's lipid in eggs, right? The egg, the yellow yolk, yeah, right. It's another one. Well, milk has it. Milk contains it. The milk fat. Right? But milk's a mixture of a whole bunch of stuff, mainly water, right? Cholesterol? Right, cholesterol is.
Like shortening or lard?
What? Yeah, oils and fats, right? That's what lipids are, oils and fats. Typically, if it's a liquid at room temperature, it's an oil. If it's a solid at room temperature, we call it a fat, although there are some exceptions, right? For example, palm oil, right, is not an oil. Coconut oil is not an oil at room temperature, right? Or harder than bread. Okay, so there are some exceptions there. But we're basically talking about fats and oils. And as you know, fats and oils are not soluble in water, right? Anyone who's mixed any kind of oil, any kind of fat or water, you know that they don't like one another, right? They segregate away from one another, right? Like being down in the South in the 50s and 60s, right? Okay? Lipids very common in the human body, right? The most common is called a triglyceride, right? Those are the most common. Okay, all the different lipids that you listed: butter, parquet, soy oil, canola oil, what have you. All the fats that you are most familiar with are what are called triglycerides. Okay, body fat, right? Adipose tissue. The lipid that it contains is, for the most part, in the form of triglycerides, right? And they're called triglycerides because they're composed of one glycerol molecule and 3 fatty acids attached to it. Okay? Your body, for the most part, uses adipose tissue for storage of energy. It uses triglycerides for protection. You have around your visceral organs what's called visceral fat, and it helps cushion one organ against the other, right? You got to think of all your organs moving around, right? Your heart's pumping, your lungs are expanding and contracting, your GI tract is doing its thing, right? And so those organs could bump up to one another and cause damage, even organ failure, right? So you have a cushion around them in the form of adipose tissue. You also have what's called subcutaneous fat, sub meaning below, fat below your skin. It helps insulate your body. So those are all of the different roles that triglycerides play in the body. Now I have an animation. I cannot remember. Some of my animations for whatever reason stopped working. They got corrupt. Okay, that's not going to work. So I can't show you a nice animation of a triglyceride, but I will show you a picture of one a little later. Okay? As I said, fats are solids at room temperature, like butter and lard and beef tallow, shortening, right? These tend to be more saturated fats, meaning they have fewer double bonds. And oils tend to be liquid at room temperature, all right? And those tend to be less saturated fats, right? Meaning they have more double bonds. Okay? Now, in terms of the saturation versus the saturation issue, right? I'm not going to test you on saturation at all, right? I like to talk a little bit about it just so you, as educated people, understand what a saturated fat is versus a unsaturated fat. All right, so I'm going to spend a little time talking about that here. Okay, so this is a triglyceride. Wish I had my video working, but if you look just at this part of the molecule, this is what's called a glycerol. So ignore everything from the carbon over, right? And you would have basically a glycerol molecule. Add fatty acids to it, 1, 2, 3. By the way, each of these little bins represents a carbon with 2 hydrogens attached to it. Okay, that's all. You have a triglyceride. 3 fatty acids, right? Everything to your left, no, everything to your right, right? Those are 3 fatty acids, and everything to the left there is the glycerol molecule. Put them together and you have a triglyceride, or triglycerol. The more saturated fats don't have double bonds between the carbons. And so their fatty acids tend to stack up very nicely. And they tend to be solid at room temperature because you have to have some heat to move those fatty acids away from one another and cause it to liquefy. That's why all the things that taste good, like butter and animal fats, right, are high in saturated fats because they're solid at room temperature. Think of all the animal fats, all the things that taste good, all the lipids that taste good that you're not supposed to eat. Right? They have cholesterol, and they also are high in saturated fats, those things that you aren't supposed to eat. And that's why they are solid at room temperature, right? Because they have this nice little fatty acids that stack up very easily, and so they're solid at room temperature. And the things that they say are better for you to eat in terms of oil, like olive oil, oils in general, but particularly like olive oil and canola oil, right? they tend to have, they are more unsaturated, meaning they have some double bonds, right? No more if they have one double bond, they're monounsaturated. If they have more than one double bond, they're polyunsaturated. And those double bonds tend to make them bend. And so they don't stack up very well, right? So they don't require heat to pull, to liquefy at room temperature. So they tend to be solids at room temperature because those fatty acids don't stack up very well. They tend to be spread apart, all right? And so that's why the oils are better for you in terms of heart disease, cardiovascular disease, that sort of thing. Okay. Now, if you were to look at the fatty acid profiles of various oils, right? At the top we have the, let's see here, red is saturated, percent saturated fat, and then you have everything that's not red is basically unsaturated, right? You can see at the top you have canola oil, all your oils up here, canola oil, sunflower oil, soy oil, right? All of those tend to be relatively low in saturated fat, and they're also liquid at room temperature. As you go down, right, to corn oil, soybean oil, particularly when you get down to like lard and palm oil, both are solid at room temperature, right? They are very high in saturated fats, okay, compared to these other oils. In terms of health, you want to be as high on this graph as possible. Okay. So again, it's nothing I'm going to quiz you on or test you on or anything like that. It's just sort of FYI. So you know when we talk about saturated fat, you know what the saturated fat is. When we talk about unsaturated fats, you know what unsaturated fats are. Okay. All right. So here's a triglyceride again. Here's the glycerol molecule here, and here are the three fatty acids. When they're combined, you have a triglyceride. Right, all of the fats that you're familiar with, your lard, your butter, animal fat, or what have you, this is the form of lipid. Okay? Everybody cool there? Okay, there is another lipid called a phospholipid. If you remember the cell lab, I introduced you to a phospholipid there. Okay? Phospholipids make up the plasma or cell membrane of a cell. All right, this is not supposed to be an egg, believe it or not. It's supposed to represent a cell. Right? That's the nucleus. This is the cytoplasm. This is the plasma membrane. And the plasma membrane is mainly composed of phospholipids. Okay, so this is our triglyceride here. All right, just going to review what we just talked about. We take a glycerol molecule, and by dehydration synthesis, we attach three fatty acids to it. We have a triglyceride. Right? Now, one thing I didn't mention, why is it that fats do not like to mix with water? Why is it that lipids don't mix with water? Yeah, Lee. It's about the polarities, right? Well, what is it about? We know that water is composed of polar covalent bonds. What do we know about lipids? Nonpolar, right? All of these carbon, hydrogens, all of them are sharing their electrons evenly. Okay? So there's no uneven distribution of charges here. Okay? Well, what does that mean? Well, that means that if there's a water molecule right here on this side of this molecule, and there's another water molecule over here, this water molecule will go around this molecule to get to that water molecule. And that's why they separate. Okay? If I'm a water molecule and Dylan's a water molecule and is a lipid, right? I'm charged, he's charged, you're not. I'm going to go around you to get to him. Right? That's why water and fat don't mix. Water's charged, they're attracted to one another, lipids are not. Okay? That's why, if you ever wonder why don't they mix, that's why. Everybody understand that? Okay? Now, so here we have a triglyceride. It is nonpolar, right, and will not mix with water. Not attracted to water. Okay? If we take off one of these fatty acids, right, and replace it with a phosphate-containing group here, we have what's called a phospholipid. Okay, all we did was take off one of the triglycerides and replace it with this phosphate-containing group. Okay, now look what we've done. Phosphate is charged. So now we have a molecule that at one end is polar, right? It has a charge. At the other end, it is still nonpolar, right? So this end of the water molecule, this end of the molecule is charged and is attracted to water, what we call hydrophilic. And this end of the molecule, where you still have those two fatty acid tails, is hydrophobic. It is not attracted to water, right? A lot of times we represent a phospholipid in shorthand like this, right, where we say it has a polar head representing this portion of the molecule, and it's got two hydrophobic nonpolar tails. Okay? Does everyone see this? Right? If you don't, let me know. I'll go through it again. No problem. Go through it again?
Yeah.
All right, cool. So, triglyceride. You understand the triglyceride? Completely nonpolar, doesn't like water. It goes poof, like spit on water. Right? Replace this end fatty acid with this phosphate-containing group. And now I have a phospholipid. This phosphate-containing group is charged. So it's attracted to waters. So now I have one end of the molecule that's attracted to water, while the other end is not. Right? Well, I don't have a picture of it. Right? Well, let's look at the plasma membrane, since you asked that question. It's a good question. How can I keep up with my chalk? Right? What would happen with something like this? Right? Let's say water is out here. Right? You see that? These heads would face out toward the water. Right? And then you have this hydrophobic interior. Okay? And essentially, that's what happens in the plasma membrane. Remember, the plasma membrane is a phospholipid bilayer. Ooh, God, love those technical terms, right? Something like this. That wasn't me. Right? Here you have, let's say you have the extracellular fluid. And here you have the intracellular fluid. Right? Okay? Because this is the side that's hydrophilic charge and likes water. Right? Do I see that? Okay. It's important to understand this because when we start talking about the cell and the plasma membrane and why some substances can cross and others can't, right? It all will come back to this. Okay? Yes, ma'am.
Why and how do we get rid of one of the fatty acid chains?
Well, there's an enzyme that comes in and cleaves that and breaks that bond. Right? Another enzyme that adds the phosphate to it.
Just because? Or like, why does it happen? Or when and why does it happen?
Well, it happens in order to make the plasma membrane unsafe. Okay. Right? It happens in the that's going to be the smooth endoplasmic reticulum is where that's going to happen. Okay. We have enzymes. We haven't got to enzymes yet. We will get to proteins, but those are enzymes that catalyze specific reactions. Basically, they set up the correct conditions for certain molecules to combine and for other molecules to be broken down. Okay. Okay. That's the second type of lipid, phospholipids. So the first was triglycerides. The second, phospholipids. The third are your steroids. Right? Your steroids. Okay? Examples of a steroid. Cholesterol. In fact, cholesterol basically is the parent compound for all the steroids that your body produces. Right? So all of the steroids originated from cholesterol. Okay? Steroids, there are steroid hormones, testosterone, progesterone, estrogen. All of those are steroid hormones. Okay? So they all are derived or made from cholesterol, right? Bile salts, which help emulsify or dissolve fat when you consume it in a meal. Okay, vitamin D. Vitamin D is a hormone, by the way, if you didn't know that. Right? Even though it says vitamin D, right? It is a hormone. Right? And it's also a steroid hormone. Okay? All right, so this is cholesterol. Right? Remember I said cholesterol gets a bad rap. All you ever get is bad stuff about cholesterol. But your body has to have cholesterol. If it didn't, then why would your liver make it if you didn't need it? Right? Cholesterol is the parent compound of all steroids that your body makes. Right? So steroid looks like a steroid. Any steroid looks like this. This specifically is cholesterol. Right? Notice it has this 4-ring structure, and then it has other what we call moieties or chains attached to it. Right? Cholesterol, steroids, as I said, they are hormones. They also play important roles in the plasma membrane, stabilizing the plasma membrane. Mentioning that. Okay, those are some of the functions. Okay. All right. So to review before we go on to proteins, lipids include your triglycerides, your phospholipids, and your steroids. Okay? Any questions before we move on to proteins? Right? Yes, ma'am.
Did you say that the steroids are all hormone derived?
No, they're all derived from cholesterol. Sorry.
Cholesterol.
Right? They're all derived from that. Basically, all of the steroids, right, are made from cholesterol. Right? You change some of these little endings here, these little side chains, and you get testosterone and progesterone and vitamin D and
I got it.
Cortisol, cortisone, right? If you look at any steroid, you're going to see that it has this basic 4-ring structure, right? Some of these things coming off of it, what they call side chains, may change a little bit, but this part will be the same. Okay, any steroid. Okay? And steroids are lipids. Right? Make sure you understand it. Lipids, steroids are lipids, phospholipids are lipids, the rest of the triglycerides are lipids. For the most part, your steroids, you're only going to obtain them from animals. Right? Now, I will not say that you will not find minuscule quantities of steroids or cholesterol in plants, but they are at such low levels that it's physiologically irrelevant. Right? The liver of animals produce cholesterol. Right? So definitely in terms of our diet, that's where we get our cholesterol. When we eat animal products, that's where the cholesterol comes from, not from plants. So
That's why we eating without animal
Well, you got a lot of things there. Your body makes all the cholesterol you need. You don't have to have cholesterol in your diet. But the issue you run into is you can run into amino acid imbalances, which we're going to talk about a little bit. You can also run into iron deficiencies. The iron from animal products is more absorbable from digestible and absorbable than iron from plant products, right? I mean, there's a lot there. Also, vitamins. There's certain vitamins that are in high concentration in animals, low concentration in plants. So, you have to watch that depending on how strict vegan you are. Right? All those things in your place. Right? So it's not one thing, it's several things. But it's not cholesterol. You don't have to consume any cholesterol. Your liver makes enough. 85% of the cholesterol in your body, your liver makes. Okay? All right, good question. All right, so are you loving cholesterol now? Right, you're going to go out and chew on some fat back. Right? Make those steroids. Chase the winner. That's a little bit. Okay. Proteins. So proteins are composed of amino acids. Right? You can sort of think of amino acids as like the building blocks of proteins, right? Alright, here comes my analogy, right? I love the analogies thing, right? If a train is a protein, then the amino acids would be the individual boxcars. All right. But the train would have to be at least 40, 50 boxcars long. All right. But that's what I mean by the amino acids are the building blocks of proteins. Right? In the same way that, say, bricks are the building blocks of a brick wall. Okay? Now, proteins have this amino acids have this basic structure, right? And I'm not gonna ask you to regurgitate this formula, all right? So I don't want, everyone going, oh my God, really? Right? No, I just sort of want you to understand what an amino acid is, okay? So all amino acids contain carbon, oxygen, hydrogen, and nitrogen. Some of them contain sulfur, right? There are a couple that contain sulfur, okay? There are 20 different amino acids, right? And the different combinations of amino acids form different proteins. Okay, so all proteins are made of amino acids. It's the exact arrangement of those amino acids that determines a specific protein. Okay? Alright, so all amino acids have this basic structure. There's a carbon bonded to a hydrogen, and then off that carbon there is what's called a carboxyl group. Right? For those of you who had chemistry, you may remember something that looks like this. Right? That's COOH, that's what that is. Right? This hydrogen can come off, that's what makes it an acid. Right? Remember, anything that donates hydrogen ions is an acid, that's what makes this an amino acid. And then you have this amino group attached to this carbon, right? NH2 is an amino group, right? That's what makes it an amino acid, right? All amino acids have this basic structure, okay? Then attached to this carbon is some type of side chain, and there are 20 different side chains, and that's how you get 20 different amino acids. For all of the amino acids, all of this is exactly the same. The only thing that differs is this side chain. Okay? Right? Here's some examples, right? So this is the basic structure of an amino acid. Here's your carbon, your carboxylic acid, your amino group. Here's your side chain, which is just represented by R. If R is a hydrogen, it's glycine. If R is a CH2COOH, it's aspartic acid, right? If it's this, it's lysine. If it's just CH2SH, it's cysteine. Okay, so you get the general idea of what an amino acid is. All of this is common to all amino acids. The only thing that differs from one amino acid to the next is the side chain. Okay?
And then again, all we need to really understand about this graph is that the base is the same and the side chain. We don't need to know these specific formulas.
Right. Okay. I did. And I took biochemistry, but you don't. Right? That's beyond the scope. It's more conceptual. I want you to understand the concept. Right? That this part of the different amino acids is different. Right? I certainly am not going to ask you to reproduce the structure of any of this. Right? I can have you do that, and you're not going to use it ever again in your life. Right? So it's pointless. Okay? Unless you're going to nutrition or something. Okay? Right? Now, I introduced this to you so that you understand that it is the side chain that determines the behavior of these amino acids. That is, some of these amino acids can form hydrogen bonds, for example, with other amino acids, right, in the same protein. Okay? So that's why it's important to understand that this portion of the amino acid differs. Okay? All right? This is just another illustration of those 20 amino acids. Again, this part is the same. Right? What's shown in here in black here is the same. These are all 20 amino acids shown right here. You see, the only thing that's different is on the side chains. Okay? And different combinations of all of these amino acids make different proteins. Collagen versus hemoglobin versus elastin versus all the other proteins that are in the body. Okay? Okay, well, I'll just go back to that. Okay, so proteins are amino acids that are joined together. Right? And you can have proteins that are thousands of amino acids long. Okay, thousands of amino acids long. typically, if they're less than 50 amino acids long, they're not stable in terms of their shape. They're just not large enough to be stable, so they change shape over time, right? They don't have a stable shape, and so they're called polypeptides. Right? So if you've heard the term polypeptides, right, they're basically the same. Chemically, they're the same as proteins. They're just shorter, right? They're composed of amino acids. They're just shorter, right? And they don't maintain a specific shape. Okay? You also may hear the term dipeptide or tripeptide. You may read about those in the book, right? The dipeptide is 2 amino acids joined together. Tripeptide is 3. All right, so I don't want you to be confused when you hear proteins versus polypeptide versus a di or tripeptide. Right? They're all composed of amino acids. It's just the number of amino acids that make up the molecule. Okay? Sometimes your book's not very, they throw around the word polypeptide and protein, and they never explain the difference. Yeah? Okay, so, all right. Function of proteins, right? Well, proteins accounts for about half of all of the dry matter in your body. That is, if you could remove all the water from your body, what's left over, half of that would be protein. Half of that would be protein. The other half would pretty much be liquid. Half of that would be protein. Okay? Proteins have functions in the body. Right? Some are structural. Some are functions that have nothing to do with maintaining the integrity of the body or a cell or a tissue. Right? But the functions are dependent on their shape. Okay? They're dependent on their shape. Okay? Let's talk a little bit about the how complex a protein shape is, just so you can get an appreciation for how important shape is. Okay? Right? So, my mouse has lost its little mind, so I can't use it anymore. Alright? So, proteins have 4 layers of structure, or 4 levels of structure. They have what's called primary structure, secondary structure, tertiary structure, and quaternary structure. Alright? And I'm not going to require you to know all these different structures. Right? Because I don't think so. I think I do. Okay? Primary structure is just simply the pattern of amino acids. Each of these little balls represents an amino acid. Right? There's cysteine, glutamate, leucine, alanine, a bunch of alanine, methionine, lysine, so forth and so on. Right? That just arrangement of amino acids is what's called its primary structure. Okay? Now, certain amino acids are attracted to other amino acids and form hydrogen bonds, and that causes that chain to arrange itself into, say, an alpha helix, or it might arrange itself into these little pleated sheets, right? And that constitutes its secondary structure. You can almost think of these amino acids as being sticky, right? And being sticking to other parts of that chain to form either an alpha helices or pleated sheets. And then you have interactions within the same molecule, mind you, between alpha helices and pleated sheets to form what's called tertiary structure. Okay? And then you can have several of these units attracted to other similarly shaped units to form quaternary structure. Right? So the shape of a protein is very complex. Okay? But it all goes back to what determines the overall structure of a protein is the sequence of amino acids, the primary structure. Right? And the function of a protein is dependent on its shape. Just like the function of a tool or a door is dependent on its shape. If I change the shape of this door, it's no longer going to be able to open and close, right? Okay? Change the shape of a key, Right? If you have a key to your house, and I say, let me borrow that key, right? I add some heat to it, change the shape of it. Is it going to work anymore? No. Right? If it does, it won't work as well. Okay? Same thing with protein. If you change the shape of a protein, it doesn't work as well. All right? Okay, so as I was saying, proteins account for half of the dry matter of your body. Remove all the water from your body, what's left over, half of it is protein. Okay? We subdivide proteins into what are called fibrous proteins or structural proteins. They're called fibrous proteins because they tend to be thin and long. And globular proteins. And they're called globular proteins because they tend to be spherical in shape, right? Like this hemoglobin molecule. Let's talk first about the fibrous proteins. Now, these are proteins that make up about 50% of your body, all right? Dry matter of your body, okay? These proteins, as I said, they're fibrous, they're long, they're thin, and they have structural functions, okay? Examples would be the most common or most abundant protein in the body, collagen, right? Collagen has a lot of tensile strength. It resists being pulled apart. So those structures in your body that receive a lot of stress, such as your bones, your tendons, your ligaments, they have lots and lots of collagen. Lots and lots of collagen. Collagen is the most abundant protein in your body. Okay? The second most abundant protein, fibrous protein in your body, is keratin. Keratin is the protein, the predominant protein that's in your hair, in your fingernails, in your skin. When you get a callus in your skin, that's where there's a buildup of keratin in your skin. Usually it's your skin's way of sort of adapting to increasing stress, right? Physical stress. I'm not saying that your skin is stressing out. Okay? Right? Those are structural proteins. Globular proteins are proteins that, yes, they are spherical in shape, but they also have a function not related to structure. Right? For example, most of the hormones that your body produces are globular proteins, such as growth hormone is a globular protein. Another hormone, insulin is a globular protein. Okay? Other globular proteins include enzymes. We're going to talk about what enzymes are. If you're not familiar with enzymes, we're going to talk about what they are shortly. Right? The enzymes are proteins. Antibodies, which your immune system uses to attack and destroy foreign particles, those are globular proteins. Hemoglobin, which is the protein in your red blood cells that attaches to oxygen, okay, those are functional for globular proteins. Okay? Now, globular proteins contain hydrogen bonds. All right, and those hydrogen bonds are responsible for their shape. Right? Now, I said that if a protein's ability to function is dependent on its shape. Change the shape of the protein, and that protein loses its ability to function. The same way as if I change the shape of a door or change the shape of your key, it no longer works. Okay? Hydrogen bonds between different amino acids in the protein are responsible for its shape. Right? Remember that hydrogen bonds are weak bonds. They can be broken if you change the pH or if you change the temperature. Right? That can break the hydrogen bonds. Okay? So, proteins can be what's called denatured. That is, those hydrogen bonds can be disrupted by a change in temperature or a change in pH, causing that protein to denature and change shape, rendering it either less effective or completely useless. Okay? All of us have seen proteins denatured. Right? How many of you have seen an egg? If I take an egg and I crack it on a skillet, put it on the oven, right? What happens to it? What happens to the egg white? It turns opaque. It originally was clear, right? And it changes color, right? It turns opaque, right? So it's white, right? That's because that high heat causes the proteins in the egg white to denature, right? They unravel, they come apart, and then it changes the physical characteristics. of the egg white, and it turns white. Okay? A good example of how denaturing a protein affects the ability of that protein to function is by looking at how enzymes work. An enzyme is a protein that interacts with molecules to speed up some type of reaction. It can speed up synthesis reaction, speed up a decomposition reaction, or it can speed up a the other reaction I can't think of. Synthesis, exchange reaction. Thank you. Right? You know. Okay. Here's a video that better work. Right? It's going to explain what videos are and how they work. It's going to explain what enzymes are and how they work. Sorry. This is a video. It's going to explain how enzymes work. Answer that. Okay, good. Here we go.
Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate.
Okay, bottom line, an enzyme is a protein. Its ability to function is dependent on its shape, right? If you go back, okay? So an enzyme is going to bind what's called substrates or reactants, right? Now notice that it can only bind those substrates if it has the appropriate shape, right? So it binds those substrates at what's called the active site, right? And it causes, in this case, it causes a synthesis reaction, like two molecules and they're being combined, right? And it releases them and it returns to its original shape. Okay, so you understand how the ability of the enzyme to function is dependent on its shape. Okay? Cool. And you also understand that enzymes increase the rate of a reaction. Right? So think of an enzyme like a matchmaker. Right? If you really want to get married like really quick, right? You don't just go into the bar scene, right? You go to eharmony.com or whatever, right? And instead of taking a couple of years to get married, maybe, match you up with someone that's like you, whatever, or not like you, however it works, right? Maybe you get married and have 2.5 kids in just a year, right? All right, but the point is it speeds reactions up to catalysts. Right? Okay, so it's a biological catalyst that increases the rate of reactions in the body, right? It's another example of that. Here we have the enzyme. It combines with reactants, in this case 2 amino acids, right? Holds them in a position that's favorable for them to bond, all right? And you get, in this particular example, you get the production of a dipeptide, okay? Enzymes can catalyze synthesis reactions. Other enzymes can catalyze the opposite, the decomposition reactions. Now, let's talk a little bit about what happens or how temperature and pH can affect the protein like an enzyme. Okay. One more thing I want to mention about an enzyme typically ends in ASE. And it typically takes on the name of its substrate. For example, sucrase is the enzyme that breaks down the sugar sucrose. Lactase is the enzyme that breaks down lactose. Many people, I think somewhere like 20% of the human population doesn't make enough lactase, right, to digest lactose in milk, right? They're known as lactose intolerant, right? Okay. So typically, if you see a term, right, from here on out in all your biology and medical classes you take, right, typically if a substance ends in ASE, it's an enzyme. Okay? Not always, right? Enzymes almost always end in ASE, but not always. And usually they take on the name of their substrates, right? But lipase is an enzyme that breaks down lipid. Okay. All right. So let's look at how temperature changes and pH changes can affect an enzyme. All right. So here we have an enzyme, right? It has a particular shape that fits the active site of its substrate. What if, due to a temperature change or a pH change, we disrupt the hydrogen bonds between different amino acids in that protein? That will cause that protein to be denatured, completely destroying the active site. Now it can't bind with the substrate, right? Just like if I change the shape of the key, it no longer works because it no longer fits in the hole and hits whatever it's supposed to hit for the doorknob to turn. Okay? Alrighty. Thank you very much. Sorry to hold you over. Well, I guess technically I'm on time. We will finish Chapter 2 Chemistry, and we will start on cells and tissues on Friday.